Wonderful. So thank you very much. This is a very interesting meeting. I always like these merger meetings between cell wall and bacterial wall biology and biophysics, because there's I mean, in the more immediate literature, there's not a lot of cross references, and so it's always inspiring to hear about other systems. So my lab is interested in bacterial cell shape, different aspects of it, predominantly working on one model system, which is E. coli, growing here as a rod, nice and steadily, maintaining its cell shape remarkably constant over time. So variations in typical shape properties are very small in E. coli and many other model bacteria, such as in cell widths, but also in length at the time of division, dry mass density, and straightness, and so on. So here, for instance, you can see how a cell that is growing as a filament, where we inhibited cell division, something we do quite often, straightens over time. So in some way, there's some sort of knowledge or memory of straightness. So the question we ask is how does this come about? How is shape robustly controlled? And so we study this question at various levels. So I mentioned or alluded briefly to the cell cycle, which determines the length of the cell at the time of division. I'll not talk about that today. Then there's the question of how cells coordinate the increase of size with the increase of other things, biomass. Is this related to pressure or density? So we've kind of heard about this a little bit today already. And then there's, of course, also the question of anisotropic growth. How does the cell maintain a non-spherical shape, like a rod shape in this case, a circumferential organization of the cell wall? And at the nanoscopic scale, there's the question of how the cell wall is organized and how the cell prevents damage, such as discussed by Marco earlier today. So I will concentrate a little bit on these questions of how, at the macroscopic scale, cells grow and how they control their shape during growth. I mean, we've seen a few introductions of this topic today. Marco gave a very nice introduction. So the cell wall of gram-negative bacteria, such as E. coli, is very thin. It's composed of long sugar or glycan strands that are cross-linked by short peptide bonds. The typical scale is like 1 nanometer, and then the scale of the pressure that expands this cell wall is of the order of 100 kilopascal in gram-negative bacteria. The cell wall is, of course, absolutely essential for cell shape and also totally shape determining. So if the cell wall is stripped off the cell wall, the cells are obtaining a spherical cell shape, and if the cell wall is isolated, it retains the shape of the cell. Then that raises the question, how does cell shape come about? So this is a process that requires growth. And so in rod-shaped bacteria, we can distinguish essentially two processes, that's elongation and cell division. So my lab predominantly focuses on cell elongation at this point. So we are asking the question, how does the cell know where and how to remodel the cell wall, how to enzymatically cut the cell wall, insert new material in order to globally control surface area, lengths, and locally maybe, I mean, it's a bit hard to distinguish these two because they're both locally and globally controlled, cell straightness and also cell widths. So I will give a bit of an overview of what we've learned in the past few years, and then expand a bit upon that, and or ask a bit of questions how we need to go about this to finalize or to establish a clear model of cell shape maintenance in gram-negative bacteria. So first of all, there's a question of how cells control the rate of cell envelope growth. And so my lab has done quite a bit of work on this recently. And we've discovered that cells actually grow grow their envelope in a way that is tied to biomass growth. And so the way, how do we discover this? So we noticed that if we measure cell density, so this is biomass density, that's the amount of cell dry mass divided by total cell volume, which is something that we measure using quantitative phase microscopy, we see that this density is shape dependent. So here, these are curves, these are averages over a large population of cells for different growth conditions. While at the same time, the ratio of the cell surface area divided by the biomass is constant across different cell shapes in different conditions. And so given that the density can be described as a ratio of cell shape, the surface to volume ratio, and this ratio between surface area and mass, we can reconcile this dependency of density on cell lengths and other shape properties, shape variables, by a simple dependency, linear dependency on the surface to volume ratio. So we found that essentially that the surface to mass ratio is constant despite changes of cell shape and also despite changes of the growth rate. So here we grow cells in a certain growth medium, in a minimal medium, and then we switch it to a richer growth medium. And what we see is that during the switch, the cell surface area here in yellow increases in the same relative manner as the cellular dry mass, which is indicated in red. So here all these quantities are plotted in relative terms, normalized by the time point of the switch. And then at the same time, the cell volume increases faster than cell surface area, which is a direct consequence of a rapidly increasing cell width, while the cell elongation rate lags behind. So in this complex nutrient shift, we can observe that the relationship between surface area and mass, or the ratio between surface area and mass, is conserved despite changes of growth rate and also additional changes of shape. So from this, we wondered how this relationship, or this robustness of surface area and mass came about. And so there are essentially two different models that can be proposed or can be discussed. So there's one that was proposed a few years earlier by the lab of Julie Theriault, which suggests that the surface area of cells increases in proportion to biomass. So this would, during steady state growth, maintain the surface to mass ratio constant. At the same time, or alternatively, there is a model that relates the surface area growth to the rate of biomass growth, which also leads to the maintenance of the surface to mass ratio. So now, in order to distinguish these two models, one can take advantage of the just mentioned or described nutrient shift. So during a nutrient shift, according to this model A, the surface area should lag behind biomass growth, such that the surface to mass ratio decreases over time, while according to this model B, surface and mass remain coupled at all times. So that means clearly from this, from the shown growth rate shift, we can deduce that among these two models, we can clearly favor this model B, and with additional shifts that we've conducted that support this model further. Okay, so that in some way demonstrates that the cell envelope growth rate is a dependent variable that depends on or is directly equal to the biomass growth rate with a proportionality factor that is a surface to mass coupling constant. So then next we wondered how cells determine their elongation rate. And so there are different proposals in the literature how elongation rate is controlled, notably by machinery that builds the cell wall. There are other types of models around. But so what we discovered, and I've essentially implicitly already shown that, is that if cells rapidly increase in cell widths while they elongate, such as in this example where cells are perturbed in cell shape maintenance using a small molecule like a drug that inhibits an essential part of the cell wall synthesis machinery, then we observe, just like in the nutrient shift that I showed you earlier, that while surface and mass still grow together here, yellow and red, the cell volume increases faster because of the increase in width, and the cell length lags behind, just like in the nutrient shift. So to us, this suggested that the elongation rate of the cell is likely a simple consequence of the changes of widths and the predetermined rate of surface growth. Yeah, and so this picture is further supported by looking at cells growing in steady state in a fixed condition without perturbations, where we observe that the elongation rate. So here, this is technically a little complicated, but what I meant to normalize all these quantities in a useful manner. So what I did here is I plot the exponential rate of cell elongation against the exponential rate of cell widening or thinning. So there's a bit of a quantity that's a little counterintuitive, maybe because of course cells don't typically increase their widths exponentially, but for the purpose of comparing these two quantities, this is what we did. And so what we see is, so here every dot refers to a single cell measurement over a short period of time. And what we see is that clearly there is a strong, simple negative relationship, correlation between the elongation rate and the cell widening. And similarly, a positive relationship between the elongation rate and the rate of surface area growth, which is in some way expected because the rate of surface area growth is simply the sum of elongation rate and widening rate. So, but on the contrary, if we look at dependencies of the widening rate on cell elongation rate or also on the rate of surface growth, we hardly see any correlation. So to us, this suggested strongly that the surface area or the elongation rate is a direct consequence of the rate of surface area growth, which in turn is coupled to mass, and to the rate of changes of cell widths. Okay, so then, as I mentioned, we are interested both in the global rate of cell elongation, but also in local changes or in the local regulation of elongation rate. And so for that, we a long time ago we teamed up. This is actually this reference is wrong. So this was in 2017. I just realized. So this is the correct reference. Lars Renner at the Leibniz Institute in Dresden conducted experiments where he grew cells initially in some donut-shaped microchambers that force these cells to grow into a bent configuration. And then subsequently, he releases the cells and studies the process of relaxation or of straightening. And so he observed that the elongation, or that the cells straighten very rapidly. So they are essentially straightening twice as fast as they elongate in relative terms. And so the question is then, how can we understand this? How can we understand this phenomenon? And that's where Ariel Amir came in, who was at the time was a professor at Harvard University, and recently moved to Weizmann Institute. And he and his student Felix Wong conducted theoretical work, theoretical modeling of the cell envelope, and suggested and demonstrated that both the phenomena of cell bending in these donut-shaped microchambers and also of straightening after release can be explained by a phenomenological model that ties the rate of elongation, the local rate of elongation, to the local areal strain, to the strain of the cell wall. Because simply due to the fact that there is pressure inside these curved shapes, there's an asymmetry in the circumferential strain, in the circumferential stress, and thus in the areal strain in these bent shapes, in these bent cells, suggesting that while the global rate of cell elongation is not controlled by mechanical forces, it has direct impact on differential growth. Okay, so coming back to the cell wall, so how is this related or how is this? How do these mechanisms? How do these observations come about mechanistically at the level of cell wall remodeling? So for a very long time, it's been thought that the rate of cell wall remodeling, both locally and globally, is essentially dictated by the rate of cell wall insertion. So this is in particular in gram-negative bacteria, where the cell wall is very thin and where any defect, as Marco showed us, can rapidly lead to lysis. So this is often referred to as a make before break paradigm, and it's supported by biochemical data, but it had never been tested at the single cell level. And alternatively, voices had slowly been raised that suggested, well, maybe the predominant role of cell wall insertion is to patch defects, while the rate of cell wall growth is dictated by hydrolases, by enzymes that cut the cell wall. So my lab studied this question by simply perturbing or inhibiting cell wall insertion completely using different types of antibiotics that are available to target the cell wall synthesis process. And so what we observed is that indeed, after a relatively short time of growth, cells lyse, as has been demonstrated by many labs in the past, and of course is the main reason for the reason for why antibiotics work, cell wall targeting antibiotics. But during this process, up to the point of cell lysis, the cells increase their surface area together with the rate of with mass in a seemingly unperturbed manner. Now you can start slow deviation of the cell volume and the cell surface area over time, which is caused by a gradual process of cell widening. But this widening on this short time scale is still very, very weak. So that means lytic enzymes are expanding the cell wall in a way that is independent of cell wall insertion. And so we've seen this with different drugs and also in different organisms such as Vibrio cholera. And that's work that was recently published in Embo Journal. So we also demonstrated that this process of controlled cell envelope growth in the absence of cell wall insertion is controlled in a very precisely and on very short time scale. So here we are perturbing the biomass growth rate over time. So we transiently essentially starve the cells for glucose using a glucose analog and then refeed excess glucose. So we can see that during this time, during this process, the cell mass, the rate of cell mass growth decreases, and so does the rate of cell surface area growth, and then they increase together, while on the long-term volume and length again start to deviate. So that means we can say that on the time scale of our resolution, or at the time scale, at the shortest time scale that we can observe of about 2 minutes in this process, these two processes are coupled, despite the fact that we inhibited cell wall insertion. So we started to have to investigate the role of hydrolases in this process, and one of the central aims of the lab is now to understand how these hydrolases are controlled and what is it that they sense, or who is essentially the regulator that dictates the rate of hydrolytic activity. And so, so far, we don't really have a good answer to this. I mean, on the opposite, what we observe is that these enzymes work very robustly. So in this experiment, what we did is we take one of these enzymes, arguably an important one, which is called MAPS, and we overexpress it strongly at time t=0, and then we observe what happens. And what happens is that very shortly after, we see a slight increase of the elongation rate and also of the rate of surface growth. But this increase is very transient, and then subsequently the cells pretty quickly settle on a new elongation rate that is slightly elevated, but by no means elevated by a degree that corresponds to the increase of the abundance of this enzyme. And similarly, if we deplete this enzyme by repressing expression and also profiting from rapid proteolytic degradation of the enzyme, we see again a very mild effect. So we see a transient reduction of elongation rate, but this effect is compensated by alternative and unknown means after a few minutes. And so by now we've started to deplete and delete genes that code for a large redundancy of these hydrolytic enzymes. And so thus far, I mean, despite deleting like 8 of these endopeptidases, these enzymes that cut these peptide bonds here, we still have not achieved to inhibit cell elongation rate completely. Okay, so similarly, actually, and this is also counter to previous suggestions. We also observed that the process of cell straightening and cell bending, that both of these processes are independent of cell wall insertion. So these are again experiments conducted by Lars Renner, and this time after inhibiting cell wall insertion. And so what we observe is that these cells still straighten. These are now treated with different drugs, just like the control, meaning the control are cells that have not been treated with cell wall synthesis targeting drugs. Yeah, so we can say that these hydrolytic enzymes have a highly underappreciated role. They are essentially the pacemakers of cell envelope growth, while cell wall synthesis still, of course, has an important role, and I'll get to that in a second, but it does not dictate the rate of cell elongation and the rate of cell envelope growth, the overall rate of cell envelope growth in our bacteria. So, and here I should say that there is some prior evidence for this. There is some a protein called curve-AB in Vibrio cholera that have been previously shown to induce bending of Vibrio cholera cells independently of major cell wall synthesis processes. This is work by Zemer-Gitay. Okay, so in very briefly, what I've shown you so far, so cell surface area growth, according to this new empirical growth relationship. The elongation rate is a dependent variable that depends on the rate of surface area growth and the rate of cell widening, which are independently controlled. And cell wall insertion has no role for controlling either rate of surface area growth or cell elongation. So what about cell width? This is where we still don't have a very clear picture yet. But so what I can say for sure is that we've observed important influences of two factors. The first one is pressure, so turgor pressure. So turgor pressure, of course, as this audience knows very well, affects cell envelope stresses directly, and so stress is directly proportional to pressure. It is thus quite possible that it affects, or not surprising maybe, that it affects cell shape. And then the cytoskeleton is the second factor. So in rod-shaped bacteria, there is a protein called MREB, which is a homolog to eukaryotic actin that is associated with the cytoplasmic membrane, lies flat under the cytoplasmic membrane, is intrinsically curved, and is oriented around the circumference of the cell. And so this, so both of these factors have a role to play in cell width regulation. So what's the evidence for this? So here, in one experiment, what we did is, in order to study the effect of pressure on cell width regulation, is we conducted osmotic ramps. So that means we changed the medium osmolarity continuously through a process that I don't describe here, to a diffusion limited ramp experiment. So that means the external concentration of sodium chloride in this case changes over time in a continuous manner. And so what we observe in this case is that the cells in response to this, they widen very rapidly. And so this widening is dose dependent. So that means if we conduct different types of ramps, either increasing osmolarity or decreasing osmolarity, we see that that width changes accordingly. So here, what we plot here is the widening rate plotted simultaneously on a different scale with the rate of external osmolarity change. And you can see that these two quantities agree reasonably well. So according to a very coarse-grained and simple, approximate and first-order model, one could say, of bacterial osmoadaptation, a well-known phenomena where bacteria restore pressure during exposure to osmotic shocks. We came up with a very simple model that suggests that the widening rate, or the rate of cell width changes more generally, changes in proportion to pressure. So while notably, and I didn't spend much time on talking about that, the rate of cell elongation and the rate of surface area changes is seemingly independent of pressure on short timescales, which is in stark contrast to what we've learned today about plant cell growth, where pressure has a very important role on regulating the cell or plant growth on the timescales we are interested in here. So then let's look at the MREB actin. So as I told you briefly, so MREB actin is forming these filaments as sketched here that are sitting or that are attached to the cytoplasmic membrane and that are directly linked to a multi-protein complex that inserts cell wall material into the cell wall and does this in a processive manner, as has been shown by multiple labs in the past, including myself as a postdoc, where we showed that MREB actin rotates around the cell on a timescale of a few minutes. So these are filamentous cells here imaged in structured illumination microscopy. And so thanks to the work of Ethan Garner and also many others, Jan Löwe and multiple other labs, we have now a pretty clear understanding that these MREB actin filaments, that they autonomously orient themselves around the circumference of the rod-shaped cell because of an intrinsic curvature preference, and that this in turn might stabilize or contributes to the stability of rod shape. So, and indeed, if MREB actin is depleted, as I showed you this movie before without explaining it much, but in this what happens in this case upon treatment with a small molecule A22, MREB actin filaments are depolymerized. We see a gradual increase of cell width, and then if this drug is washed out, the cells restore width. So with these types of curves, we can now start to entertain models of how MREB and also pressure contribute to width changes. One short note I want to make here, without showing any modeling yet, is that the increase of cell width and also the decrease of width here are gradual. So that means the rate of change of the rate of width change increases slowly. And this is in contrast to what we've seen upon the application of osmotic pressure, where width changes very rapidly and the rate rather decreases over time. So this suggests to us that the effect of MREB on cell width control might be indirect. And this is also supported by the beautiful AFM images we've seen from Jamie Hobbs' lab, where apparently the absence of MREB over long timescales leads to disorder of the cell wall. And we speculate or reason so far that it is this effect, the lack of order, that will ultimately that leads to the cell shape changes, and this in turn requires some time. Okay, so what I've shown you today is that different aspects of cell shape are controlled in different ways that are in some way modular, at least at first order. So first of all, the surface area is controlled by a new relationship that ties it to biomass growth. Cell width is controlled to some extent osmotically, but to an important extent, of course, also to the MREB cytoskeleton. And these two quantities that in turn affect biomass density, the quantity that we imaged in the first case, they are seemingly independent on the timescales that we're interested in here. That means on the timescale of about one mass doubling time. And then I've shown you that mechanical stress and strain affect both widths in a pressure-dependent manner, and also differential envelope growth during bending and straightening. So with that, I would like to thank the people that were doing this work, notably Enno Oldevoordel, a former postdoc, and Yuki Kitahara, with the help of many others, such as Baptiste and also Gizem, and then, of course, all our collaborators. So thank you very much for your attention. I mean, of course I have the role of salesmanship here, of course, right? But I would definitely say so. I think, I mean, this came as a pretty big surprise, and I think if you consult the opinion of expressed in reviews, it's the focus over during the past 10, 20, 30 years has been predominantly on cell wall synthesis, and it's the hydrolases are only slowly, or by now they have gained a lot of traction, but they're also harder to study, so that makes it a little difficult in some way to make strong advances in this regard. But I think the focus has been very much on cell wall synthesis, and of course cell wall synthesis is an important process that is essential for cell survival and thus prime target for antibiotic therapy and so on. But its impact on cell shape and cell shape regulation, I think, has been overestimated.
So Sven, can I ask you, when you were saying you were deleting up to 8 of these, so what does that mean in terms of total percentage of hydrolases that are usually present?
So there are more than 30 different enzymes that cut different parts of the cell wall. When we deleted these 8 and leaving 1 under inducible control, then those are all known endopeptidases to date. So these were described by Manjul already, and they are and also by others. And so in these enzymes are essential. So that means the cells do not survive on the long run. But on short time scales, if we deplete the remaining enzyme, and so I must say, I must admit here at this point, we need to still confirm that we really deplete this to the level, to an undetectable level, essentially. Then we see continued elongation.